So thanks. Yes, yes. Oh, sorry, we're responding to a message. So yeah, hi. Thanks for joining us in the seminar. I'm Christopher Cousins. I'm a PhD student in Tiffany Garcia's lab here at Oregon State University, and it is my honor and privilege to present the speaker for our graduate student nominated Ecology, Evolution, and Conservation Biology seminar series, Dr. Gabriela Parra Olea, professor in the Institute of Biology at the National Autonomous University of Mexico in Mexico City, UNAM. Dr. Gabriela received her PhD from UC Berkeley, and her research is on the study of amphibian diversity and conservation with a focus on the systematics, evolutionary biology, and conservation of amphibians in Central America. She has described 20 species and 2 genera of salamanders in Mexico and has identified at least 30 other species that are still in the process of being described. In addition to being at the forefront of conservation work in Mexico, Dr. Gabriela is also part of the International B-cell Task Force, a collection of experts from around the world that work together to prevent the spread of the pathogen B-cell. Thank you for accepting our invitation to speak, and I'm looking forward to your talk.
Thank you, Chris. Well, first of all, I have to say I haven't spoken English in a very long time, so I probably will try to mix Spanish and English words in my presentations. I mostly know them by heart, but in Spanish. So sometimes I'm gonna stumble a little bit, but thank you for the invitation. And the other thing you have to know is that due to COVID, I out-exiled myself in Quintana Roo, which is in a very small town called Mahahual. It's right on the coast. It's beautiful. The reef is incredible for scuba diving, but because it's a very small town, the internet it's very slow. So I asked everybody else in the house to stay off the internet tonight, but if something happens, just let me know. I know my students are there. 2 of my students are there. I saw them already, so they can just quickly text me or something and say, you're losing me or something. So now I'm gonna share my screen, and please tell me if you see me. If you see and hear.
Gotcha. Yeah, it's good.
Okay. Let me, do I have, okay. I want to do this full screen. I'm sorry, I'm very slow, so with technology. I can't do this full screen for some reason. Oh, it's here. There. Can you see?
Yes.
Yeah, looks good.
Okay. Can you hear me all right? Perfect.
Yes.
Okay, so I'm the worst with titles. I always give the same title. So I have, if you see my CV, there is this title like every year because I never come up with smart titles and I'm just lazy, and I usually write them like in the traffic lights, and it's always the same. So, but this is actually this is my main line of research, biodiversity and conservation of Mexican salamanders. But when I talk about biodiversity, it's usually phylogenetics and systematics. And when I was looking or preparing this talk, I decided against it because it's not as interesting as conservation. And so today I'm just gonna talk about conservation and mostly the projects that are happening right now in my lab. How do I move? Let me see. Here. Okay, so I got my PhD at UC Berkeley with David Wake, like seems like a million years ago, but it's only about 20. And then I spent 2 years at Harvard University working with Jim Hanken, and I focused on the study of Mexican salamanders because basically David Wake was the only person who had studied or had paid any attention to Mexican salamanders at the time. So this is my group. I still work with both of them. We still describe a lot of species of salamanders, and we still have plenty more species to be described. The photo in the lower part of the slide is half of my group. And this is where we went to the field, we were collecting, and this blurry thing here is a dog who couldn't stay still for 5 minutes while everybody else was on a frozen smile, waiting for the picture to happen without a flash, of course. But I just wanted to point out that this is not the whole group, but about half of them. And tonight is, because for me it's past 7 o'clock in the evening, but today I'm going to be talking mainly and mostly about Delia and Angel's research, and I'm going to use the slides, and I can already say thank you, Delia, and thank you, Angel, for giving me their presentation, so I can tell you about it, because it's really fascinating. So, you all probably know amphibians, and you know that there are 3 big groups of them: frogs, salamanders, and caecilians. And this is the numbers for Mexico. There are about 300. I'm gonna stop every time my computer says your connection is unstable, because I know you stop hearing me. So, and then I'm gonna repeat the last phrase. I'm sorry about these inconveniences, but so in Mexico, there are about 397 species of amphibians, and we come to be the 5th richest country in terms of amphibian diversity. And you can see here in this graph, I hope you can see my pointer, and most of the diversity of Mexican amphibians is arranged or is composed in 2 big groups: hydrid frogs here and plethodontid salamanders, which is my main focus. My PhD, I did my PhD studying plethodontid salamanders, and of course they're my favorite and they're beautiful, but I also like frogs. And caecilians, we only have 3 species of caecilians, but they're also fascinating. Okay, so unfortunately, amphibians are going extinct. There's a huge crisis in amphibian diversity of amphibians, and there's something called the Global Amphibian Assessment, who is an organization, it's not an organization, it's a group of us organized to evaluate the conservation status of all the species of amphibians. And most of them have been evaluated all over the world. So in general, in terms of the world diversity of amphibians, there's about 6,000 species. And there is over 30% of them that are in drastic decline or that have gone extinct. 1,800 species are in critical danger of extinction, and with 122 already declared extinct species. For Mexico, the IUCN gives the category of risk, and we recently re-evaluated most of the species. And as you can see here, 24% are critically endangered and 21% are endangered. So between these 2 categories, we have almost 50% of the amphibians in Mexico under critical danger of extinction, which is it's a very high number. There are many reasons why amphibians are declining, and unfortunately, we cannot point to one of them exclusively. In some places, there's one reason, in other places, there's another reason, and most of the times, there's just the synergistic effects of many of these factors acting together. So climate change, of course, we're all very familiar with the effects of climate change, introduction of exotic species, overharvesting, logging for wood, and the emerging infectious diseases. And tonight I'm going to tell you about how the populations of salamanders have changed over time, and I'm also telling you about the emerging infectious disease that is affecting amphibians all over the world. How do we know that the populations are crashing? Because we have historical data, and this is where it becomes important, where the collections or the voucher specimens deposited in collections become super important, because we know they were there and how many of them were there. So we can compare the collections, the samplings in the 1970s and current samplings, because we have the data from David Wake's studies in the 70s and the 80s. So they were the first to come and study plethodontid salamanders, and what they did was to establish elevational transects. Here you can see 4 in Veracruz, Oaxaca, Chiapas, and Guatemala, and describe the species diversity along these elevational transects. And as a result of these studies, they found many new species of salamanders that were described in the following years. Now we have that data because they were very careful. They didn't have a GPS at the time, so they were very strict with getting the data from the field sites and describing their collections, their sampling. And so they wrote how many individuals of every species they found and where they found them, under a log, in the tree, in the bromeliad. So we have all of that data from the 70s and the 80s, and then we can compare them to the recent samplings that we started doing in the 1990s when I started my PhD here, and even to the last 2 years where my student Angel started working there. This is how the transect looks like. This is all 4 of them go from the sea level to almost to high elevation paramo. Here, this is the Cofre de Perote, one of the transects where Angel was working. And as you can see here, there's like in every change of vegetation, like here to go from this forest to cloud forest to pine and oak forest, you can see in every change of vegetation, there is a different community composition of salamanders. So these species, like these 5 species of salamanders that occur in the lower pine and oak forest, they don't occur anywhere else, basically in the world, not even in Mexico. Many of them, some of them have wider distributions, but most of the plethodontid salamanders in Mexico and in Central America are microendemic. So you find them in this little range here and nowhere else. So as you can see, there are almost 20 species in this transect of about 60 kilometers that goes from the coast to the high forest paramo, high elevation paramo. These are Angel's results after sampling in the 1990s, was when I was doing my thesis, and Angel started doing 2016 and 17, and they all show a collapse in their populations. He has these 4 localities in that transect that I showed you. They have abundance declines of over 85%. So you can see how these species, for example, in this locality, Barranca de Teocelo, was abundant or somewhat abundant in the 70s, in the early 70s, and it declined in the 79, in 97, basically it was gone. In the 2016, he didn't find anything. And the same pattern we see in La Joya, in Las Vegas, in Las Lajas. Basically, all the populations of salamanders collapsed after the 1980s. Here is the structure of the salamander community and how it changed also again in time. This lower left graph is 1979 to 1981, where Wake and his students found 14 species of salamanders. And in 2016-17, Angel found only 9 species. And it's really interesting to see how all of these species were lost. 5 species are gone from these localities, but the habitat generalist, Pseudorhizia leprosa and Achilorhizia cephalica, became way more abundant. So they didn't collapse, and they are doing actually much better than they were in the 1980s when they had to share the habitat with other species. There's plenty of these graphs. So this is Pseudorhizia smithia, Pseudorhizia unguidentis in Oaxaca, in the second or the transect in Oaxaca. And you can see also 70s, they were really abundant. The Pseudorhizia smithia was super abundant. See how many species they found, how many individuals they found in one sampling season, more than 200, and then nothing. So the collapse was very strong and very evident in many of these localities all along the country. This is like a general graph with many localities where we have Oaxaca, Veracruz, and these arrows show the collapse in the 1980s. So there is a clear pattern of amphibian, or at least plethodontid salamander communities collapse in the 1980s. This is the result of the survey, which I started in the 1990s, and now Angel redo the same surveys in 16 and 17 for his master thesis, and then for his PhD, and we find this dramatic decline in salamander abundance. Some pseudorhiza species were hardly hit, and the more generalist microhabitat generalist species, they're doing really well, or they stayed well, or they're more abundant than before, while the habitat specialists are going extinct. And we observe these declines stronger in the mid elevation and the high elevation forest. But if you remember the graph, the lower elevation populations have less diversity of species. And we find that species that occur in the lowlands adapt quite well to banana plantations and things like that, because as long as the humidity is retained, they find a microhabitat where they can survive. Now I'm going to tell you about emergent diseases, which we all very familiar now, thanks to COVID, so we all understand how they work. But just a general introduction, emergent infectious diseases are those that have appeared recently or that have existed in the past, but have recently increased in frequency, geographic scope, or both. Infectious diseases are caused by the presence and activity of one or more pathogenic agents, including virus, bacteria, protozoa, and other multicellular parasites. The mechanism of transmission is very simple. It's just the convergence of one host and one parasite within an ecosystem. And I listed a few of these diseases for you because you're familiar to this. The mosquito-borne infections like dengue and malaria, the ones that you can get through bad food or contaminated water like cholera, the ones that are transmitted by ticks, such as the Lyme disease, and you can also get infections by mollusk, infected food, and other mosquito bite other insect bite, like leishmaniasis. You probably have heard them, all of them. More familiar to us as humans, there is the plague, the cholera, and the malaria, the flu, and our new disease, which is COVID-19. And as I understand, it's very dramatic, and it's really difficult. It's been really difficult to us as humans or to survive through this, but I wanted to show you that it's not the worst that humanity has survived. And the death rate, at least the last time I checked, was about 2% of the population was dying out of COVID, with a total of 2.5 million deaths worldwide as of today, because I checked in the morning. But look at here, for the plague, in the 14th century in Europe, it killed between 20 and 45% of the population. It was really horrible, right? Cholera, 13% of the population in the 1830s. Malaria, even nowadays, between 600,000 people die every year out of malaria. And the flu, it's the first, one of the most famous flu, because we've had several in the 1980s, called the Spanish flu, left more than 40 million deaths. So COVID is horrible and we're suffering from it, but it's not the worst that we have seen. So we'll survive. Okay, I think. There's a map of the many diseases for humans all over the world. There's probably plenty more, but we're just not aware of them because we're not living them. Well, only COVID, but here's what happens everywhere else. For wildlife, which is the focus of our talk, we also have plenty. So, for example, prairie dogs get this. This is the year of emergence of the big catastrophic effect. Prairie dogs have monkeypox, civet cat has SARS, birds with the West Nile virus, small rodents, hantavirus, non-human primates, AIDS, and small rodents, Lyme disease. A map again of the wildlife diseases in birds, mammals, reptiles, and fish and corals, which is now it's drastic also what's happening to the corals. But just so you see, there's plenty of diseases out there. There is this fibropapilloma, which happens in the sea turtles when their immune system is depressed. They get these things, and they also get the lungs affected and the internal organs, so they die out of it. The white nose syndrome is a fungus that affects the nose and the wings of the bats. They can also die. They have many of them die every year out of this disease, and they have also episodes of big collapses. This is Spanish. I'm sorry about this writing in Spanish, but I just wanted to show you how all these are factors that are tied to emergent diseases or the events of huge mortalities, and they have to do with these changes we're doing in the ecological system, like agriculture, the water use, extra logging, climate change, and how we are making it worse as humans by how much we move around the world and how easy we move around the world. We're seeing it right now. We have in Mexico, we had one strain of COVID, and now there is this England strain, and now the South African strain, and we move with it. We move the disease with us all over the world. So that's a big problem. And climate change is making things very, very difficult and very hard to understand also, because we never know if due to climate change, the immune system of the organisms got depressed or the virulence of the pathogen became more aggressive. In amphibians, we have these virus, bacteria, and fungus that have caused emergent infectious diseases. In my lab, we work with fungus, and just as a reference, there have been collapses of virus and bacteria every now and then in different populations. So the virus, we have two kinds, IRIDOVIRUS and RANAVIRUS, and there have been collapse of amphibian communities due to one of these viruses. Here is a global distribution of the RANAVIruses over the world. So most of at least here, I don't think we have sampled very much. For bacteria, we have these Aeromonas, which affect also the tadpoles, and they call the red-legged disease, which you probably have seen if you've gone out to the field to look for frogs. You've seen these red-legged specimens. Some they have caused a massive die-off, but have not caused, as far as I know, bacteria and virus have not caused the extinction of any species. And there's the focus of our study, which is the fungus, which is this fungus is the disease is called chytridomycosis, and is caused by a chytrid fungus. And they have observed massive die-offs, and this is Delia's slide, but you can see here hundreds or thousands of dead organisms by this chytrid fungus. Now, the chytrid fungus is the chytrids is a large group of fungus that are parasites. They are all parasites of algae, plants, nematodes, every other organism. They occur in all of the habitats. They occur in the water as well as in the substrate. They have aquatic zoospores with a flagellum, so they can move, so they can disperse large distances. And what happens with this chytrid fungus, specifically in areas of the skin of the adult or the oral disc of the tadpole. Here is a mouth, the tadpole mouth, which is a healthy one. And here you can see the fungus grows in the keratin part of the mouth, so they die because they can't eat. And in adults, when they get it, they get it all over the skin, so they become lethargic, they lose their appetite, they stop moving, they start shedding a lot of skin, and they die, basically. Not all of them, but as I can tell you later. So how did this amphibian decline crisis started? It all started in 1989 during the Third World Congress of Herpetology, where many people presented the talks and started saying they have seen or they have witnessed a huge collapse in a population or a massive die-offs. And this is normal, it happens in all of the species, all these massive die-offs every now and then, but it was just too interesting to notice that it was happening at the same time in many parts of the world. So it became a pattern. It was not like isolated events of massive die-offs. The amphibians were dying all over the world by the thousands. So they got together and they organized a workshop for the following year, 1990, and they started doing research and they started doing field work to find what was the reason of these massive die-offs. It took 10 years to figure out that it was a fungus. Here's the name, Batrachochytrium dendrobatidis, which we call BD for easy, was described in 1999. So it was a long time, you guys. 10 years is a long time to figure out what was wrong with these populations. And then not too long after, there was a second species of chytrid described, Batrachochytrium salamandrivorans, in 2013. So when everyone started looking or finding their populations collapsed or dying or the massive die-offs, all of these questions during the Congress and during the workshop in the following year they were stated, like what's happening? Why is it happening? Why is it happening now? How is it happening? Is it a parasite? Is it a new parasite? Is it the climate change that the immune system of the amphibians got compromised and suddenly everyone's dying? But then not all of the species were dying, right? Many were dying, but not all of them. So why was that? And so just to be brief, it's been 30 years since it all started, and we know a lot. We don't know everything, but we already know a lot. This is a map of the occurrence of PD, and you can see all of the black dots. It's presence of PD, and I can tell you basically it's everywhere you look for it. There is a coincident mass extirpation of amphibians in the Neotropic with the arrival or emergence or re-emergence of this fungal pathogen, which happened in the 1980s, as you can see this in the graph, 1970s, 1980s, at least in Mexico, southern Mexico, and Central America. And not all of the species reacted in the same way, but this is really interesting. Here you can see North America, the severity of the decline, but look at Mesoamerica, which includes Mexico. A lot of the species got affected, and South America as well. So not all of the species reacted the same way or went extinct or got affected by BD. Some of the species, just like us humans with COVID, some of us get infected and nothing happens. Other species immediately died, and other species got a decline in abundance, and then they recovered. So there is this variation in response to BD. The next thing they wanted to know, or we wanted to know, is who is BD, right? Is it one strain? Is it the same strain killing everyone in the world by dispersal, or is it different strains that became virulent at the same time due to other factors like climate change? And so it took a few more years, see here, 2013, to be able to get a lot of genetic data to propose a scenario. And they found that there are multiple lineages of PD. There is two, I think now there's 5, but there is 2 very abundant or widely dispersed, the GPL1 and GPL2. There are some endemic lineages like the Brazil lineage, which is endemic to Brazil. Some of these lineages are very old, up to 100,000 years, have been in the area coexisting with the amphibians. And others are arrived by dispersal, right? There have been cases of hybridization where one strain arrived and hybridized with endemic lineage and caused or created a hypervirulent strain. This is more or less a distribution of the two big strains. GPL1 is the blue one. GPL2 is the red one here, and a little bit here in the US. And BD Brazil, well, is in Brazil, and it got introduced here, I think, through the trade. This is more or less a distribution of the strains so far. Now, so what do we know? To answer these questions, what was it? It's a fungus. Why? We don't quite know yet. There's still a lot of research going on. How? Because it affects physiologically the specimens and they die. When did they become abundant? They become in the 1970s and 1980s, and they are directly linked to these amphibian declines. Is it a new parasite? In some places, yes, it arrived by dispersal. In other places, there are endemic lineages. What is the point of origin? Is Asia. It was, they were able to pinpoint the origin where it comes from. Was it due to climate change? We don't know yet, but maybe. And was it the immune system? Well, it's probably linked to the effects of the climate change, but it has not been like completely proved. Why some species and not others? Because there is very complex relationship of everything in the organisms with everything in the environment and everything related to the parasite itself. So it's not very easy to say one single thing. There's this complex relationship. Now, that's for the world. Now, for Mexico, we have the same questions. What is the history of it in Mexico? Was it introduced? Or was it native and it dispersed to Central America? When did it arrive? It was introduced. Which strain we have in Mexico? And why are the observed declines in the 70s and 80s linked to the arrival or the reemergence of PD in the country? Well, we started doing surveys all over the place, and for now we can say that there's more than 70 amphibian species that have come positive for BD. They all have, most of them have very low incidence rates. And we find them everywhere, as I said, everywhere you look, you find BD. We have found them in the deserts in northern Mexico, and also in the high elevation places. So it's everywhere. Was it introduced, or is it native, and when did it arrive? And for this question, Delia and Angel went to the voucher specimens collected by the Berkeley team in the actually even earlier than that. In the 1970s was an interesting period to sample because there's a lot of salamanders collected at the time, but it's a big collection. So they sample all the way to the 1890s. And Delia found that the earliest record of PD in Mexico was in 1894 in Baja California. So with that, we can say that it's not introduced, at least not newly introduced. And here in this map, you can see the incidence. We're looking for the red dots here, which is the BD positive, 1894, and then in the 1930s, in the 1940s, 50s, 60s, and 70s. Of course, it became more and more abundant as the years passed, right? But it's been here for a very long time. Okay, but this is the results from Angel. This is the Cofre de Perote transect. And here you can see the red colors here is BD positive. And in those localities, he did not find them before 1979 when he starts seeing these collapses of the populations. So I have here some graphs. You can see here, this is the graph that I showed you earlier about the abundance decline, over 80% of the population decline, and the appearance of PD in these red lines. I hope you can see them, but here is Barranca de Teocelo, and the same story happens in La Joya, Las Vigas, and Las Lajas. So he finds a direct correlation of the population collapse and BD presence. He did not find them before these years in these populations. Now, which strain of PD is present in Mexico? And for this question, Delia sampled all over Mexico. Here is the red dots are the museum swabs because we wanted to look at the BD presence of BD genomics from the voucher specimens, which turned out to be really, really difficult and almost impossible to do, but she still sampled them. And the green dots, it's the current sampling. So what happens is that she went to do next generation sequencing in a collaboration project with UC Berkeley. But in order to do this, you need more DNA than the one you get from a swab. So she went to the field, collected specimens, took a small sample of the skin, and she tried to raise the strain of PD present in the organism. So she did that, and then she went to Berkeley and to do the molecular work, and this is what they found. There is one strain, the yellow, the colors here represent different strains. There is the common strain, the yellow one, is the same as GPL1, the one that was very frequent in the US. And there are two cases of GPL2, which is the one from South America, one in Baja California and one in Chiapas. So there basically is one dominant strain in all over Mexico. Unfortunately, it was really difficult to get the genetic data of the voucher specimens. So this data is mostly from recent collections. We don't know yet, she's still working on it, but we don't know yet the sample from the 1890s, which strain was it? Because this one, see, so uniform, looks like a rapid dispersal of one strain in Mexico. It could be, there could be more than one, right? One of the ancient or the endemic lineages could be just being taken over by the more generalist GPL1 strain. Okay, so the conclusions for this is there was a dramatic decline in salamander populations in the 70s and 80s, at least in Veracruz, in Eastern Mexico, associated with increase of BD prevalence. BD is present in Mexico since the 1890s, and there is currently a widely distributed strain, which is the GPL1. The GPL2 strain is present only in two localities, Baja California and Chiapas, possibly also as a result of a more recent introduction. We would love to have more genetic data from the historical samples. And the species we see today are the ones that survived, basically, the ones that survived the epidemic, and they live with low BD intensities now. When I introduced the chytrids, I told you about two species, Batrachochytrium dendrobatidis, which was the original, or the first one described in 1999, and the second species, Salamandriborans, which was described in 2013. This time, it was the same beginning. In 2010, 96% of wild mortality in the Netherlands. The salamander populations just collapsed. And because we have all of this knowledge acquired from BD, they quickly moved and sampled everything and isolated everything growing in the skin. They found out quickly it was another chytrid, and they sampled basically all over the world to see where it came from, right? So the time frame goes it begins in 2010 with 96% wild mortality in the Netherlands. In 13 and 14, they have wild mortality in Belgium, and then it starts appearing in salamanders of the trade in the UK in 2015, and then in Germany. So by 2016, it's present in the Netherlands, Belgium, Germany, in the wild. They, as I said, they sample all over the world, and they found that the point of origin was Vietnam, Thailand, and Japan, and from there it moved through the trade to Northern Europe. And it's now present in, it's present in wild salamanders in Asia and present in museum records in Asia of over 150 years ago. So it's native to that area. A big problem comes with Batrachochytrum salamandriborans because unlike, so from BD and the studies of BD, we can say that it's very dangerous in mid-elevation, cloud forest, and high elevation, but mostly mid-elevation forest because of its optimal growth in laboratory, which is here, from 17 to 25 Celsius. This is the temperature. And salamandriborans flourished in the Netherlands with much lower temperatures, between 10 and 15. So this puts at risk all the salamander populations that occur in high elevations, which is a lot of species, but not only the tropical region of Asia. So the span of optimal growth is huge because it goes from the tropics, from very high temperatures in the tropics, up to 10% Celsius, which we find in the high elevation populations, right? Why is it a threat? Because also, unlike BD, it seems to do better or to target or to be more dangerous to salamanders. And here is they tried to see how many species of anurans of amphibians reacted to B-sal. And here in the green one, we have a lot of frogs, and here's all this clade is frogs, and they all came out resistant to B-sal. But this clade is salamanders. So this little clade here came out as resistant, this one too, but not here. Look at all of these species in red, is all of the salamandridae family. They all died from infection of B-sal. And why is it a threat? Well, why should we care? Well, because we should care as humans, but also because between US and Mexico, we have over 80% of the salamanders of the world. So we should care if B-sal arrives in the continent. There are two risk models, like the environment, the B-sal suitability for North America. North America includes Mexico to us. And here in this model, they use the data from the localities where B-sal was found in Northern Europe. And they find that in Mexico, there is the suitable habitat is here in the Trans-Mexican axis. But this is Delia's, this is Delia's projection, and she uses the data, the ecological data from the populations in Northern Europe, but also the ecological data from the point of origin, which is tropical Asia. And this predicts many more areas in Mexico as suitability, with very high suitability for B-sal. After she did that, we looked at the salamander richness by quadrants all over Mexico, and we find these hotspots or these areas with very high diversity of salamanders to put them together with the areas of high suitability for B-sal and high salamander richness. And we proposed all of these units of management of all the hotspots where there is a very large species diversity and very high B-sal suitability, that we should be worried. I mean, we should be worried about everything, but we cannot protect the whole world or the whole country. So we need to focus on smaller, more manageable areas. And this is what she proposed. Now, for North American species, This is the original paper by Martel, but then some other species of North America got also tested, and as with every epidemic, we find species that are not affected at all, some that are affected but have subclinical effects, and others that die immediately out of the exposure to Bisol. And out of the 23 species tested for North American salamanders, 7 died. 3 plethodontids and 4 salamandrids. And in these plethodontids, we included 2 species of Mexican salamanders, and only 2 because we don't want to bring the strain to our laboratory. I really worry about it spreading out. So we took the salamanders to the US, and it's a big problem with customs and everything. And they also, the salamanders get stressed. So you have to wait a large quarantine for the salamanders to relax and then test for BD and then infect with Bisol and see if they die. Anyway, the 2 species that we carried to the US, or that Becky Hartman carried to the US, died quite quickly out of exposure to Bisol. So there is 1 high elevation species here and 1 mid elevation species here. The low elevations don't survive the movement because they just die. So it doesn't look good. It's really worrisome. We didn't wait as long, or it didn't take as long as with BD. So there is a group of people working, which is called the Bisol National Task Force. It's a union between Canada, US, and Mexico. We're really super worried about Bisol arriving in the country, in the continent. And so there's a lot of research on how to mitigate trying to find ways to mitigate the effect if it happens or when it happens. And here's just a few pictures of the research. They keep the salamanders in small containers under controlled temperature and everything. And this slide shows how the amphibians move through the trade. And this is what needs to stop. We have to stop having amphibians as pets, especially if they come from a different country, because they're bringing all of these diseases with them. And it's very high, the risk if Bisol comes through the trade into the continent. It's just going to be a matter of a few months or a very short period of time for it to be all over the place, all over the continent, just basically killing all of the salamanders. And I'm going to stop there because I have learned from my son that he cannot pay attention for more than 30 minutes, and it's been 40. So I would like to thank my people, because I'm just very fortunate to have them working with me. And this tiny person here, the other ones are my students and friends, and they do all the work. Basically, I sit in my lab and I just stare at my screen. They go out to the field, they do all of the laboratory work, and then they do all of the paper writing. And I'm very happy to have them as students, and very proud to have them as students, because they work with each other a lot and they help each other a lot. For one student that needs to go collecting, there's always a van full of people willing to help. So I'm very happy about it. And thank you for your patience. And if you didn't understand something, there's Chris to translate, or there's Delia. I saw her in the audience. She can take questions if you need more details of her analysis. And yeah, that's it for me. Okay, stop share. Okay.
Awesome, thank you, Dr. Gabriela. We did have some questions come in. So we can start, I think the first is from Tiffany Garcia. And she asks, she says, thank you, what a fantastic seminar. Thank you for the comprehensive look at amphibians at Mexico. I'm curious why BD seemed to have such a large impact on salamanders in Central America and Mexico. In North America, our salamander species are largely tolerant to the BD strains here.
Yeah, so as I was saying earlier, I think BD was more aggressive in the mid-elevation forest. So the cloud forest, the species that occur in the cloud forest got really hit really hard. And also the high elevation pine and oak forest, but mostly cloud forest species. And I think it has to do with the conditions of the environment that makes the perfect environment for BD to grow and to be more effective on their host.
Okay, we have another question from Andrew Blaustein, who says, nice seminar. We have found differential sensitivity to Bacrochytrium among populations of the same species in frogs. Have you seen this in salamanders?
No, we have, well, what happens, talking about BD, right? What happens is that most of our species are microendemic. And if they got hit by BD, basically the whole species went extinct. And in the species that we have more with a wide distribution, like Pseudorysa leprosa and Pseudorysa cephalica, they occur in many different habitats, and they all have very low intensities of BD. So no, we don't see that. But we haven't compared very many populations of the same species, mostly because the distribution range of all of the species is very restricted, and the ones that have a wide distribution, they all have very low prevalence of BD.
Thanks. We have another question from Jason Jones. And I think this is referring to the elevational surveys that were done, comparing the historic and then the more recent data. And he asked if there is a chance that these differences between the historical and current data could be attributed to different survey methods or effort.
I don't think so, because it was David Waite who sampled in the 70s and 80s and his students, and he started sampling with me in the 1990s. So we have the same school. And then Angel resampled recently, which he learned again from us, and he went to UC Berkeley and studied all of the field notebooks. And they were very detailed of how many hours they spent in the forest and how many specimens they saw and did not collect, because in some of the places where they found hundreds of specimens, they only made a note. They didn't collect them. And Angel compared only the times where, like the rainy season, right? Because Berkeley people came in the rainy season, in the dry season, and we collected also in different seasons. But he only chose those times where they were actually comparable and adjusted for the number of people and the number of hours to do the collecting, to do the sampling.
Okay, thank you. We have a couple more. Nick Van Gilder asks, or he says, thank you for the great talk. I'm wondering if you have any estimation as to how much more amphibian diversity in Mexico still waits to be discovered or described.
I mostly worked with plethodontid salamanders, because that's how I started working, and there was before very few people interested in working on the systematics of amphibians in general. So I focus more on plethodontid salamanders because there's a lot of work. So for plethodontid salamanders, I can say that we have at least 20 species to be described, and we are very conservative because we have described the EC species, the ones that you can find genetic divergence, morphological differences, ecological differences. But then we have a whole genus like Thorius, which are miniature. All of the species are miniature, and they all look the same. So they're very difficult to describe. And with those, we are all more conservative. We're not describing as many as there could be. So, and for frogs, I have no idea because I haven't started. I have a couple of students who have started working on frogs. And every species with a wide distribution is normally a species complex, but I don't have an estimate for frogs. I can only say that confidently for plethodontic salamanders, at least between 20 and 30 species to describe.
That's a lot of species. We have another question from Cynthia Dinora, who asks what conservation actions you propose to reduce the collapse of salamander populations.
Yeah, well, in terms of Bissau, if it comes to the country, there's nothing we can do about it. Because if it disperses through the communities, there's no stopping, I don't think. But if it's not about an emergent disease such as BD or Bissau, then we can preserve the forest. I think we need to educate the people, which some of my students, Alex, who's here in the audience, I saw him, he started teaching people about the importance of amphibians, the importance of keeping the amphibians, and they teach the children. They have all of these outreach, right? Which we have not done, basically. I have not done outreach, but Alex, he started doing outreach for the ambistomatid species. And I think it's really important for people to know how important amphibians are for the ecosystem. And this is only going to happen through education and through the preservation of the forest, because if they log down the forest completely, well, there is the habitat of the species. The species go extinct. So I think we need to educate our people, and we need to show them how to preserve their forest, not just for the salamanders, for the whole forest, because climate change is, an evidence of one of the things that are happening due to our logging or poor management of the ecosystem.
Okay, thanks. I think we have looks like we have one more question, and I think it's kind of a follow-up. It's from Stanley Sessions, and he asked, with the association of Bissau with the pet trade, he asked if the likely vector from the captive populations to the wild populations is humans.
If we are vectors?
Yeah.
Well, no. I think we can spread Bissau by having contaminated water or by having poor management of introduced species. But I don't think that we can, if I understand correctly the question, I don't think we can be infected by Bissau. We can carry it through other, like probably the nets, the water, the boots, if we're sampling in a contaminated place, or if we have infected specimens, salamanders at home, and we don't clean the water, and that water goes to the drainage, then we know that the drainage ends up miles and miles and miles away from our home. But I don't think that humans, we can be a vector. I don't think so, but.
Yeah, I think that that piece that you just mentioned about dumping the water is like really important, because I think that's something that not a lot of people could think about.
Mm-hmm.
But yeah, well, thank you so much. I think that about wraps up questions and everything. And thank you again for making the time and accepting our invitation to come and speak. It was great. Thank you.
Well, thank you, Chris, for having me, and thank you everyone for staying a full hour.
And Tal, did you want to do a plug for